Great. So thanks to those of you who have joined us so far. So this is the repeat session, and this is so welcome actually to the very final breakout session of the RDA plenary. Some might have been very long 2 weeks for some of you, but hopefully we'll still get a bit of work done in the last session of the plenary this year. So welcome to the FAIRification of Genomic Annotations Working Group meeting. This is the official initial meeting of the working group as an RDA plenary, since it's been recognized as a RDA working group. So the title of this meeting is Metadata Harmonization at Scale. And we just go on to the next slide, please, Sveno. So as you know, the session will be recorded. So we'll be, as with all the sessions at the plenary, we'll be recording this session. So just to introduce myself quickly, my name is Ryan O'Connor. So I'm working as part of the RDA Tiger project on RDA Europe, and I'm working with Sveinung, Anna, and the other co-chairs of this working group as part of the support we're providing the group from Tiger. So I've just pasted into the chat a link to the collaborative notes document. So there's a link to the slides and other various bits of useful links and collateral materials in that document. There's also a sign-in sheet towards the top of the document there. If you could please, there's a table in there. If you could just please fill that in with your details, that would be much appreciated. I'll just run through the agenda in a second, but in the meantime, if you could please keep your microphones on mute. We'll have a couple of presentations coming up, but there will also be quite a lot of time towards the end and at various intervals for any questions you might have. So we'll invite you to unmute your microphones and come forward with any questions, or if you have any questions throughout the session, please feel free to add them to the chat. Next slide, please, Sveinung. Thank you. Yeah, so I'll just go very briefly through the agenda before I hand over to my colleagues, Sveinung and Anna. So first of all, we'll just run through a brief introduction to the various concepts around genomic annotations. We'll then have a series of lightning talks so the first of these will be from Assistant Professor Anna Bernasconi, who is one of the co-chairs of the working group, who is here with us today. Then hand over to Dr. Sveinung Gundersen, who is another one of the co-chairs, who's also present in the room today. Will then be followed by 3 recorded talks from Nathan Sheffield, Adam Wright, both of whom are co-chairs of the group, and then from Dr. Thomas Brown, who is working at the Leibniz Institute for Zoo and Wildlife Research, who will be offering a perspective from the biodiversity background on the potential impact of this group's work. We'll have a short section for any questions or any kind of clarification questions you might have after these presentations at that point, and then we'll be handing back over to Sveinung, who will give us a bit of an overview of the working group's work plan, and then we'll be opening up the discussion to any questions that you might have around the specific use cases that the group is planning on working on, and the various tasks that the working group has set out in its work plan. So that is it from me for now. So what I'll do is hand over to Sveinung. Please, Sveinung, take the floor.
Thank you. So I'm going to start with a brief introduction to what we mean by genomic annotations. So my name is Sveinung Gundersen. I'm a senior engineer working in Elixir, Norway, at the University of Oslo. And I'll come a bit back to my background later in the lightning talk here. So I'm just going straight for this. I'm starting sort of at the very beginnings, because there might be people here that do not really know life science or bioinformatics. So we're starting at the very basics here, really, and what the genome is, and go from there. So what we are talking about here is at least one term for it. This is genomic tracks. And this is a unified model for data analysis, and I'll tell a bit more what I mean about this. So starting from the genome, the genome is basically the totality of DNA of an individual, and that can be expressed as a sequence of letters, one for each of the 4 nucleotides. So it looks something like this. In itself, it's very difficult to get any meaning out of that, so you would need some way to annotate different parts of the sequence with more information, and that is basically what we're talking about. That's the data that we're talking about here. So to make sense of a genomic sequence, you need to annotate it with some data elements that, for instance, relates to functional processes. So one basic, and I guess the initial example of that is the position of genes. Where in this sequence are the information that's translated into proteins, basically. That's the genes. But that's not the only type of information. There's lots of other different types of information. And basically this is, you can look at this as a sort of one-dimensional data model where some sort of information can be put on a sequence line. And the sequence line sort of forms the coordinate system here. Yeah, so in conclusion, genomic track then is a series of related data elements positioned along a reference genome sequence, forming one sort of a line which is showing information about the same type of element. So this concept was created in the context of something called Genome Browser, which is a visualization software where you can then look at different region of a DNA and then look at these different tracks depicting different information types and figure out if there seems to be interesting things happening across there. But it's not only for visualization. So there's also a lot of different analysis tools, some of which are visual, some of which are not, that make use of this kind of information. So even though this is sort of a visual model, it's also important as a analysis model. Yeah, and then there is very many different types of data that can be expressed in this way. So I mentioned genes, and there's lots of other things referring to either the function of DNA or binding or structure and the way sort of DNA is compacted and variation and all of this. So I'll not go into detail, but there are some examples here. Yeah, so that is tracks. And then there's another term called genome annotation, which is also used in some contexts. And basically, that is the initial characterization of a newly assembled genome. So when you create a sequence, you typically run this through some sort of pipeline that finds the basic elements like genes, like regulatory elements, and so on. So in a sense, this is the sort of original type of track. And I'll come a bit back to why it's sort of these two different ways of denoting what's more or less the same type. Yeah, so when we started working with this working group, we tried to figure out what should we actually call this, and we looked into many different possibilities. There's the Scandinavian proverb saying, "A dear child has many names," and in literature we can see this being described in many different ways. So we came up with a suggestion, which is the genomic annotations, which is actually, we mean it a little bit different than genome annotations. It's a bit broader. And there's some support for it, but it's not really that much used. So it's a bit a new thing, but at least we found that this would be the best description that we found for the totality here. Yeah, and the reason there is this sort of a bit of split in nomenclature is that also there's a bit of split in the research field here. So on the one side, you have fields like functional genomics and epigenomics, which is mostly human biomedical research, but also other model organisms. And typically, you're then working with the same coordinate system. You have one sequence or a reference genome, and then you analyze and look at many different types of tracks on the same coordinate system. And then there's typically the actual genome annotations are created by some sort of central authority, either a consortium or repository. And then on the other hand, you have more the biodiversity and comparative genomic fields, where the focus is much more on the plurality of genomes, mapping the tree of life. And from a purely data-oriented point of view, then you have different coordinate systems. Typically, they're trying to compare across different sequences. So things looks a bit different. And basically, you would need to also distribute the generation of annotations across different projects and so on. And yeah, so that's a bit of the basis for that. So what are we trying to do here? What's the reason that we created this working group? So there has been a lot of different large projects costing with data, public data that's costing billions to produce over the years. And typically they end up with some sort of data portal that where you can download and use this data. Unfortunately, they all have a bit different metadata models, and also the APIs and the ways to download things are different. So there's been a need for harmonization of this metadata for many years now. And then on top of that, there's the thousands of smaller scale data sets, and many of those also have little or poor metadata. And this is more on the biomedical side. And in addition, as I mentioned, there's a lot of biodiversity projects now, most of them under the umbrella of Earth Biogenome Project. And the goal here is to produce millions of new genomes, sort of mapping the biodiversity of nature. And all of them need to have some annotation, and there is a need for harmonization of the metadata and also the distribution of those annotations. So to sum this up, in 2 questions here, can we harmonize and FAIRify genomic annotation metadata across all of these projects and beyond? And can we build a simple community-oriented infrastructure for discovery and reuse of genomic annotations? And that is sort of the scope that this new working group will try to handle. So now we will move over to the co-chairs presenting lightning talks, and also Thomas Brown, who represents the biodiversity community here. So I'll leave it over to Anna. Here.
Okay, thank you, Sveinung. I will share my own slide deck. Okay, so I'm sharing. Do you see the right?
No.
Sorry.
Yeah, you see the wrong screen.
It went nicely the time when we tested it, and of course, okay. Yeah, it should work now.
Yep, looks fine for me.
So I'm going to present you this general talk about building integrated and fair repositories for genomic data. And actually, I will spend most of the time on the Data-Driven Genomic Computing ERC Advanced Grant project that has been led by Professor Ceri in Politecnico di Milano. I worked under his supervision between 2016 and 2021. The project has as main outcomes 3 main abstractions: a genomic data model, which is quite similar to the concepts that Sveinung just presented. So it is very much in line with the idea of FAIR tracks annotations. A geometric query language, which is basically a way to use and query compose complex biological queries over these tracks, and then a genomic conceptual model, which is for superposing a layer of semantics on the metadata. So a few words on the genomic data model. Basically, it means dividing the genomic data into data sets. Each data set is composed of different samples. Each sample is basically a collection of regions. By regions, we mean that concept that Sven also called in many other ways, also annotations or tracks. Basically, each region is an interval in the space of a chromosome with a start and end position and the strand. And to this region, we associate information on the experiment that has allowed to measure these regions and some key value pairs metadata. Thanks to this representation, we can then perform very complex queries. Here, for example, we are representing on somehow genome browser line of promoters, 3 of peaks according to chip sequencing and mutations. And here there is an example of this query language that we developed to from genes, we extract the relative promoters, and then we can do a cover operation, which means that we only keep those peaks that are confirmed in at least 2 tracks. We do a distal join based on the promoters and the peaks that are closest. We do a map, and then thanks to this operation, we can keep only the promoters that are located in a region where at least 1 mutation was located. So all of this is enabled by the representation of genomic tracks as interval-based regions. And finally, we had the genomic conceptual model that overlays a conceptual layer, a semantic layer on top of the samples. So we describe metadata also along the direction of biological information, the donor, the biosample, the management view, the extraction, and the technological view. Thanks to this representation, we were able to build a big repository that started from very well-known genomic data sources, transform them, clean information, map them into a relational database, enrich them with the ontological information, and did some checks. So the bird's eye view of the project was to enable bioinformaticians, biologists, geneticists, not to have to query all these different sources, but having a unique interface called GenoSurf on top of the repository, where you could and extract the regions all in the same format, and therefore use them in the GenQual system, which enables these complex biological queries. And very briefly, I would like to mention also a current project that we're leading now is the Better Real World Health Data Distributed Analytics Research Platform. Here, as Politecnico di Milano, we are focused on leading the work package 4, which is in charge of doing the health datasets verification and preprocessing. We're partnering with about 15 other institutions, and the ultimate goal is to empower healthcare professionals with the exploitation of large sets of multi-source health data, building a decentralized infrastructure. Very briefly, here is the schema. So we are in charge of building the pipelines that go from the raw health data to the FAIR databases. So we'll have all the information of the hospitals in the same format, and therefore this is the representation that will be accessed by these trains that host the machine learning models that learn from this data and can answer complex healthcare questions. And with this, I close here. Thank you very much for your attention.
So then I'll continue sharing my screen. So where do you start this again? Here. Yeah. So I'm going to talk about a project called FAIRTracks and also mentioning a software product called OmniPy here. So my background into this started as a developer of a software called Genomic Hyperbrowser, which is a framework for doing statistical analysis on this type of data, the genomic tracks. What we discovered then is that we needed a simple way for people to search and discover and import data, and that wasn't really available. And we tried different ways of doing that. Tried first sort of having our own collection of tracks, but then we tried with different ways of importing, and then we found out that there was a need to do some harmonization of the metadata to some level. And that sparked a project called the FAIRtracks, which was then an Elixir implementation study. So that this was together from Elixir Norway, together with Elixir Spain and the EMBL EBI, and specifically Track Hub Registry team there. And I think this happened more or less in the same time frame as this project that Anna described, but I was not aware of that, and the scope is quite similar. So at least we're working together now. So that project ended up with a published set of recommendations, and also the services that we produced and the data model that we produced got awarded this mark badge called Elixir Recommended Iterability Resource. And then we wondered, okay, we do need to take this to a more global level and get more users and more traction around this. And then we got in contact with the RDA and the RDA Tiger project, which is then facilitating RDA working groups, and was selected as one of the pilots there. And then the last years we're sort of building up to this and getting more people interested. So that's the background also of the working group and the FAIRtracks project. So I'll say a bit more what we did in that project. So as I mentioned, we published recommendations on how to adopt the FAIR principles for genomic tracks, and identified, looked at all the different pieces of the different principles, and mapped that to this domain in quite some detail. So that's in the publication if you're interested. And we also defined a metadata model. It seems that this is quite in line with what Anna's project defined. So that's one of the things that we will do quite soon, start to harmonize this. So this is then designed for as a minimal metadata schema. So it's not meant for sort of storing every metadata that you can store for in a for instance, experiment, but what is most needed for discovery and reuse. So it's a minimal set. And it also connects to other services from Elixir, FAIR enabling services. I'm not going into more detail here now. We also implemented a series of services connected to this data model. So there's a metadata validation and augmentation service. We implemented the search service, and which also integrated to the Galaxy framework. Yes, and then we have also in the later years looked more into how this would translate into the data lifecycle, and I'll not go into detail here because there's many elements, but basically how we are looking at this is that this would be probably a secondary cycle after the typical data lifecycle that's been in use now. Then we are looking at the secondary cycle, which focuses to harmonize and improve the FAIR aspects of the metadata. We also developed a website, fairtracks.net, which is in its basis designed to function as a community website. So it has quite a bit of information about tracks and browsers and data collections, FAIR concepts, and so on. So currently it's focused on the outputs of the FAIRtracks project, but it can easily be transformed into a hub for the FAIRification of Genomic Annotations working group, and that's at least my intention here. And then I'll also mention a Python library called OmniPy, which has been developed now the last couple of years, basically in anticipation of this working group, because it's obvious that there's a large need to harmonize the existing metadata. There's lots of metadata out there, and in order to create a system that can easily support the wrangling of that metadata into the proper harmonized shape, and create some pipelines that can be rerun when there's changes either in the model or the schema, or I mean that's the model or the data or ontology terms and so on. So this OmniPy library is a way to specify data models as Python classes, and provides powerful features for parsing, validation, conversion, and interactive use of that, and also connects to orchestration backend that you can use to run these pipelines on compute infrastructure. This is still a bit under development, but it's very much moving towards an actual functioning product. It's also, I can mention, it's also domain in specific, so we can use this for completely other things than the genomic annotations. So these are the people that have been involved in the FAIRtracks project, and these are the groups. So I'll leave this over to you now, Ryan, to run the other pre-recorded ones.
Perfect. Thanks, Anna. Thanks, Sweinung. So yeah, as I said, we have 3 pre-recorded lightning presentations, so each of about 5 or 6 minutes in length. And then we'll, if you have any questions for sort of to Anna or to Sweinung, any clarification questions on those presentations, or if you have questions on either of the 3 lightning talks that possibly we might be able to answer, or that we can kind of forward on to the presenters, then please feel free to add them to the chat or come forward. So I'll just share my screen. So the first of the lightning talks is from Associate Professor Nathan Sheffield, who is one of the co-chairs of the group. So I will just press play, and if Sveinung or Anna, if you could just nod if you can make sure that we can hear the sound. So let's press play right now.
Hi, I'm Nathan Sheffield. I'm an associate professor at the University of Virginia. My lab studies human epigenomes, and we have a special interest in breaking down barriers to data access and use. Today I want to describe a project we're working on that addresses some of these challenges with epigenome data access. The project is called BedBase. It's basically a database of bed files. So we're trying to catalog all the published experimental genomic region data sets that we can find, put them in a central location, and make the data more usable. So an important goal of BedBase is to make the data easier to find and easier to access. We're working on a web interface, command line client, as well as Python and R packages. So these different interfaces would allow you to retrieve, cache, and analyze the genomic region data in a variety of ways, interactively, as part of a tool if you're a tool developer, or in a reproducible script, or whatever you need. So what's the problem that we're trying to solve? Well, as you probably know, human epigenome data has been exploding. There are now almost 100,000 bed files available on the Gene Expression Omnibus. So it's great that there's so much data available, but the problem is that it can be hard to find. Maybe even more importantly, it can be hard to access and use, because the interfaces aren't really set up specifically for bed file data in terms of making them easy to use interactively. So to give you an idea of what I mean, here's a little video where I'm searching for some data. So I look for Ewing sarcoma, ChIP-seq data. This is a cancer that I'm interested in. If you search for this, you can find some data on GEO, and you get a list of possible results, and you can identify some ChIP-seq data that you're interested in. So this particular example has some ChIP-seq data, and the way you could find it, get access to it, the actual files themselves, you look down here in this custom menu, and here we have these narrow-peak files. And narrow-peak are files that are identifying genomic regions, similar to bed files. You could select a few files of interest and then download the data, and this is basically going to download an archive, a tar archive of those files, which you can then look into the archive and then grab the gzipped narrow-peak files for the different samples of interest. So this is sort of fine, but it's really geared toward a human. Less easy for a computer to do. Now, if you want to look at metadata, you have sort of a similar protocol where you're going to use this FTP server and use the FTP server to download a compressed soft file. And then you can open up the soft file, and at the end of the day, you're going to end up with metadata that looks something like this. So a lot of really useful information in here, but in a format that's a little bit difficult to parse and use in your downstream analysis. So it's great. GEO is great, and we use it all the time, and it's an excellent source of data. But it can be a little bit difficult to use and to get access to the data. So I'm going to show you a little bit of the alternative of what we're thinking with BedBase. So we're working on, first of all, a search interface that's going to use semantics to make it easier to find what you're looking for, but I won't talk much about that now. If you've got a specific bed file that you're looking for, you're going to get a view that looks like this, that gives a lot of information about that bed file immediately. Because BedBase is specific to genomic region data, we can build interfaces that are really geared toward this data type to show you things like the number of regions and what is the distribution, as well as a bunch of other at-a-glance information to help you know if this is going to be the file that you're interested in. In addition to this, everything's API-oriented. So the point is that this is programmatically accessible. All the different statistics that we've computed about the data, as well as some analyses results. And you can also use the API to get the bed file itself, or a compressed version in a big bed file. In addition to this, there's we're making this open API compatible, so there's documentation for all the API endpoints, and so that's basically a brief overview of what BedBase is. So the goal here is to provide a really nice interface for this really important data type, so that tool developers and data analysts can make better use of the huge resources of genomic region data that already exist. So this is why I'm interested in our new Genomic Annotations RDA working group, in which we're going to be developing community standards around how the metadata for these files should look, and hopefully also get to do a little bit of work around how APIs like BedBase can communicate with one another to make it easier for all of us to use this really important data type. So thanks for listening. I hope you will give BedBase a try as we develop it, and feel free to drop me a line with any feedback or ideas. Thanks.
So thank you in your absence, Nathan. So the next presentation is from, this on full screen, is from Adam Wright, who is another co-chair of the working group from the Ontario Institute for Cancer Research. Just in advance, I'll just let you know there is a slight buzz on this, kind of buzzing sound in the background of this. The sound of this isn't great, but it is legible, so it might just be a case of having to turn up the volume on your own device. But I'll just press play right now.
Hello, I'm Adam Wright. I'm a technical manager on the Reactome project. I'm also 1 day a week on the Alliance of Genomic Resources. I've been in the field for over a decade, and I one of the side things that I like to do is work on coordinating metadata and metadata standards, and I'm a co-chair on this working group. I'd like to talk to you today about two different topics that have to do with one another. JBrowse genome browser and a reference genome header specification. And it will become clear, hopefully, as I go, why I'm presenting on these two things, and then I'll explain my proposal for what we could potentially do with this within this working group. So JBrowse itself is a fairly well-used, widely used genome browser that anybody can install. It's open source, and you can configure it to have your own tracks. You can pull in tracks from others, from their instances. You can have whatever reference genomes that you would like. You can have DNA or protein sequences. It has a lot of different visualizations. One of the things that I want to focus on with this is that when you click on the name of the track to the right, there's 3 dots there. You can click there and get more information on that track. So that's where you would get the metadata for the track, which is similar to what this working group is working on. So with all sorts of genome browsers is the intent for this working group, but I want to focus on JBrowse. I think that it's a good starting point for this sort of work. So another thing that I have published earlier this year with a group of collaborators is a file header specification for reference genome files. Normally, FASTA files don't have a file header. The format of ours is the same as YAML, and then but with semicolon tilde after it, denoting that this is the header. Normally, in the original FASTA specification, the semicolon denoted a comment, so we're taking advantage of the comment line. Not all tools will run with this, but it's a great way to put information in there, and we find it to be minimally invasive, being that if you want to run this through a piece of software that can't ignore these comment lines, you could simply take out the header. We provided tools for that, or you can do it whatever way you like. But having ever increasing number of reference genomes it is creating a need for having these sort of specifications. So here's the configuration for JBrowse that I talked about earlier, where you can, within the JSON file, you can specify for a track the metadata file that you're using. It will show up as text within the JBrowse interface. I'll show you that on the next slide. But for each track, you can specify metadata, and there is no standard right now, and the standards created, or the specifications created by this group, could be used and recommended to users to use, and tools could be made to help them make metadata for all of the different tracks that follow FAIR principles. So here's one of the places where this has already been taken on is within the Alliance of Genomic Resources. For each one of the species, there's something like 6 species now have joined the project. They all have JBrowse instances, and a lot of the mods will have many JBrowse instances for each one of the reference genomes. And so within these JBrowse instances, we have added, well, Scott Kane in particular that's managing that. He has been creating headers that follow our specification that show up within the browser. And yeah, this is all configured through that configuration file in the previous slide. Very simple, but gives the user a lot more information than just like the information above that provides information on the based on the file name. And so yeah, what I'm proposing is that we create a tool, say in Python or something like that. We could agree upon that, that goes and validates the JSON configuration file for JBrowse to make sure that it follows in the specification that this group comes up with, and so information get pulled from somewhere to make sure that the information in the tracks are fair, or also just it could be a validation tool that it follows a certain set of rules that we come up with within the group that could come up with later, but it's a simple project that could have a reasonable impact. That's it for me. Thank you for listening. Bye.
Thank you, Adam. And finally, we have a presentation from Dr. Tom Brown from the Leibniz Institute for Zoo and Wildlife Research based in Germany. So Tom isn't a co-chair of the group, but we got in touch with him, well, Sveinung got in touch with him and asked him to provide a quick presentation from his background in biodiversity and on the potential impact that this working group might have. So I will hit play now. So as I said earlier, just if you have any questions, maybe you have some clarification questions for Anna or Sveinung, or for the other presenters that we might be able to answer, but if not, we'll certainly forward them on to the presenters later. So just pressing play now.
Hello, my name's Tom Brown, and I'm the coordinator of the IT and Infrastructure Committee in ERGA, or the European Reference Genome Atlas, which is itself the European node of the Earth Biogenome Project. And I'm going to be giving you the biodiversity perspective on genome annotations and some of the roadblocks we're facing in terms of publishing, using, and reusing them across the tree of life, and how we've started the process of trying to make genome annotations a fairer process in terms of producing them, publishing, and reusing them in the community. So the EBP and a number of regional and other nodes have the ambitious aim of sequencing and producing reference quality genomes for all eukaryotes on Earth. And this has resulted in an explosion in the number of these assemblies being published, but the annotation of these genomes, and in particular the publishing of these annotations, is lagging behind. And I've shown this simply in this graph where I've plotted in black the number of cumulative reference genomes per year, which are deposited in GenBank, and below those in blue, which contain a RefSeq annotation, or in red, a user-submitted GenBank annotation. This really highlights that thanks to technological software and the sheer democratization advances of the process of reference genome generation, we have a huge number of genomes at use, but not many that have public annotations which can be freely exchanged via databases such as GenBank. And this is a huge issue because a large number of the downstream analyses which we want to perform rely on the annotation of a genome, and in particular, being able to use the same annotation for analyses in different research groups. So the reason why this is the case is complex, but one of the consistent issues we fall into is inconsistent file formats, and in particular, different databanks accepting different file formats. So ENA and NCBI both accept different formats themselves if one wants to deposit an annotation in their databank, either MBL or table format. And this itself can only be deposited by the assembly team. So both an annotation and the genome assembly can be packaged into an MBL or table format and submitted to ENA or NCBI. These file formats themselves are not generally used by researchers or software which they would use to analyze genomes. And in particular, if a third-party research group wants to publish an annotation, this cannot currently be done. And this is a huge roadblock in terms of sharing annotations once published with other researchers. So currently the state of play is that normally research groups will create ad hoc annotations, doing the best they can with methods normally shared in papers or briefly written in databanks. And this has created a large reusability problem among the field. However, we're hoping this will improve in the future as the community is starting conversations with both in ENA and NCBI to accept both a file format which is accepted by the field, for example, GFF3, and one that contains the necessary information in terms of understand what it contains and how it's been made and by whom. And hopefully soon these databanks will accept such annotations in part of their submission process. Within ERGA, we're currently working in an EU Horizon project called Biodiversity Genomics Europe to produce and annotate around 500 reference genomes for species found in Europe. Not only are we able to publish these annotations, but we're also working to develop annotation workflows and benchmark them across as many species as possible in the tree of life. We're working closely with Ensemble to make sure these are of a high standard, but also Workflow Hub to be able to publish our workflows written in SnakeMake, Nextflow, or Galaxy in a format with full software and versions, and can be redeployed by anyone with access to either HPC or with the infrastructure provided by Galaxy. And I encourage you to read about the efforts we did to benchmark some of these pipelines at the Biohackathon last year and follow our Workflow Hub space where we'll be publishing our workflows in the future. So in terms of what we would like to see to make an annotation fully fair, we would like primarily a file format consistent with the software that we use. This is typically GFF3 or GTF, and accompanied with this, a full documentation of the pipeline, software and version, the data and databases used to annotate such a genome, and the genome version itself. We hope that with all of this information, we'll lose this need to duplicate efforts and computational cost of re-annotating genomes many times whenever a research group wants to analyze a genome. Finally, I would encourage you to get involved with ERGA if you would like to discuss more with the community about anything involved in the production of a reference genome, from sampling to the bioinformatics to even ethics and policy. You can register on the link here or follow us on any social media. Thank you very much.
Okay. I'll just close that down and stop sharing my screen. So I guess, yeah, thanks. I know we're continuing now but thanks to all for the presentations from the co-chairs, and especially thanks to Tom Brown, even though he isn't here, for contributing that recording. So I don't see any questions in the chat. So I did say if people have any questions for either Anna Sveinung or any questions on any of the other presentations, we do have a few minutes now if they'd like to pose them, or we can move on. As I said as well, I'm happy to take them in the chat function in Zoom as well, if people are more comfortable with that. Okay. Sveinung, I guess the next point on the agenda is just a quick overview of the just looking a bit more closely at the working group itself. We've obviously had some of the background information and some of the projects and activities that have led to the formation of this working group, but I'll hand over to you to give a summary of the work plan and just where we are at the moment.
Thank you. Thanks a lot. So I'll start sharing again. Yes. So overview of the working group work plan. So as a bit of an introduction to this, so first I'll say a bit of about the use cases and then how we are aiming to solve those. So as a bit of a background here, this is one of the slides that I made a number of years ago and been using for a number of years in context of this analysis software that I mentioned, the Genomic Hyperbrowser. And basically, we were thinking of the sort of data space here in terms of a matrix, hyperdimensional matrix, where you have sort of a reference genome sequence coordinates in one directions, and then different types of tracks or information, and then you can sort of add on dimensions on various metadata aspects like cell types or tissue types or types of data and so on. And basically this sort of forms a hyperspace where you can do different types of analysis in different dimensions. So that has been sort of a concept that we've been having for a number of years, and I went to a presentation last year in the GA40H context from a very impressive presentation from Anshul Kundaje, and basically you see the same kind of illustration here. And he was then doing AI-based methodology, collecting lots of data sets and doing this sort of very impressive analysis on that. So there is definitely a need that's been there over a number of years that we're seeing being realized more and more now. Which leads me to the first use case here, which is to support discovery and collection of datasets to build AI models. That's sort of the headline here. I can read in detail how we have written it in the case statement. And this is the main focus here would be biomedical analysis, but also other types is definitely in scope. So the discovery of genomic annotations through harmonized metadata to improve the availability and scope of datasets for analytical applications, including methodology-oriented software tools and AI/ML efforts. So that's sort of the more biomedical use case. And then the second use case that we have defined is a biodiversity-focused one. Title here is genome annotations in biodiversity as FAIR objects. So how we have defined this is that we want to establish genome annotations for biodiversity assemblies as FAIR objects with improved metadata and integration with relevant infrastructure and tools, including both manual and automated annotation of genome assemblies. And this, as Tom Brown here described, is very much trying to provide solutions for needs that is very much in the community. And just to mention that we are not aiming here to take over the role of the publishers, but more to provide a community-driven alternative where we can sort of independently, at least at first, provide metadata and publication of that metadata for genome annotations in a way that's more agile and probably more easily able to meet needs and possibly can be adopted by the larger repositories if they're interested in that. Which and we have been talking to them, so there seems to be interest. This is also meeting a need that's been described from the Earth Biogenome Project. There's a report on annotation standards last year where they very much identify a need for metadata standards for annotation, which is lacking. The third use case is more about distribution of the datasets. So there is this existing infrastructure called TrackHub infrastructure, which is a solution for sharing annotation data or track data, which is in use at least with the two major genome browsers, which is the UCC genome browsers and Ensemble. Yeah, I can describe this. So we want to enhance the genome browser related infrastructure for TrackHubs with harmonized metadata and related services to simplify the process of generating, hosting, and registering metadata enriched TrackHubs. And metadata is the key here because typically there's minimal, if any, metadata being shared in this way. So adding metadata to all this will also have quite a bit of impact. Yeah, so that's the three main use cases that we are aiming to provide a solution for. And this is an overview of the working plan, the deliverables that we aim to implement or develop that will help realize the possibly a solution to these needs. So the first of this has been mentioned several times. That is a recommendations for a minimal FAIR metadata schema. So we want to create a data model harmonize the different models that have been described here, the Gecko data model, the FAIRification model. There's also International Human Epigenomics Consortium. They have a data model with representatives in the group, and there's also other data models out there. So we want to harmonize those and to extend those with fields that are needed by the different use cases in different communities. And then on the top of that, we want to build a infrastructure around this to make this not just a data model, but also usable for services and then the end users. And we are not, so we are sort of building the recommendation or the structures around this. And the first important part here is to provide some recommendations on how to build this transformation pipelines, because that is we assume then there's some metadata in some core form somewhere, and we want to harmonize all of that, and that's where the sort of title of this session also comes from, the metadata harmonization at scale. So this will be a big part of the effort. So we want to first create some recommendation on how to make those pipelines. And yeah, so basically there is lots of different data sources that we want to harmonize from, and there's also a set of different common normalization and mapping processes that we would like to generalize and scale up and provide some recommendations for how this can be maintained. And all of this is very much in motion, all of the aspects here. So the data might change, the data model might change, the ontologies might change, and so what. So building something that does not grow stale, that's the challenge here. And then we would like to provide some recommendations on how to persistently store this, to publish this using some public repositories in some way, so that it can be discovered and harvested by different services. And connected to that is the issue of identifiers. So we want to make it easy to identify a particular track or annotation. One aspect that we might also look into is the content derived. Is that possible? And this is the idea is based on a related working group or standard coming from the Genomic Alliance for the Global Alliance for Genomics and Health, which is now producing a standard called sequence collections, and Nathan, who's a co-chair of this group, is also then the leader of that working group. And basically that is about providing identifiers for the genome sequence, which also Tom Brown mentioned was a need. That is content derived. So if you have the actual sequence, you can then generate an identifier from that. So the question is can that similar concept be used also for genomic annotations? So that's something we will look into. I don't know if we have time to really solve that, but that's at least a question to look into. And the last part here is as mentioned by Nathan, is some standardizing of the APIs for search, because we are not here in this group implementing the actual search engines, but we want to allow different types of solutions for that. But in the end, if those end up with different APIs, then we're not really solved anything. So we need to standardize that so that downstream users or downstream tools can use the same API for searching, independent of the providers of the search engine. And then the third deliverable here would be then proof of concept integrations with third-party services. So of these recommendations. So the actual sort of development will be done a bit outside of the scope of the working group itself, but by other funding, but the coordination of this will be definitely inside of the working group scope. And the last deliverable is then to develop some resources that support community building and adoption. And we want to then build a sustainable community of people with passion for data integration in the life sciences. We are aiming for a broad representation with people with contexts, background from data producing projects, and people from the FAIR and RDA or RDM community in general, tool developers, domain experts, analytical end users, and any other people that's not on the list here that are interested in joining forces. So this is aiming to be broad community. Yeah, and then the deliverables here would be the actual sort of structure, web pages, documentation, and so on to help build and sustain this community. So at the end there, I just also wanted to talk a bit about collaboration possibilities or adoption of other recommendations. Here's a list of different working groups or interest groups in RDA that we think might be relevance or might be some overlap, and we very much want to adopt the outputs other people have made, so that we don't have to develop everything from scratch and end up with with sort of double work here. So we are very much aiming at extracting the existing knowledge and adopting that for our purpose. So if you are in any of these groups, or if you have some other groups that think might be relevant, then please let us know, and we're very interested in collaborations. I can also mention, as I also sort of alluded to here, or I said straight out, there is a connection to the Global Alliance for Genomic and Health. Mentioned sequence collections working group. There's also a study group on experiment metadata, which is of very much relevance that we want to adopt here, and also another one on sequence annotations, which is also highly relevant. So we also hope that this would help foster collaboration between those two organizations. Yes, so that was what I had on the working group. So now we have set up some time for a discussion or comment section. Put up some focus points here, which are suggestions. So I can just list them. So any challenges within the use cases, any comments on the individual tasks, or possible intersections with other RDA groups and efforts elsewhere, or any thoughts or issues or questions or comments you might have, which is outside this. So yeah, I'll stop sharing now, I think.
I'm just put into the chat there just for reference. So obviously we've kind of discussed the main aims of the case state of the working group so far, but I've just put the first version of the case statement in the chat. So obviously there's a bit more detail on the deliverables, etc. Just one thing to note: there has been a slight update on that since we submitted it. So that's the very first version that we submitted for community review, which that period ended a few weeks ago. There's been very minor changes to that, but the sort of main the main body of content in there is still relevant, and we're just waiting for the final, so that RDA Council endorsement before we publish the last version. Anyway, so yeah, if does anyone have any questions or comments or any sort of clarification questions on anything that we've covered so far today? I'm conscious that we do have a bit of time for open discussion, but that obviously depends on any questions that any of the attendees might have. Or Anna, if there's anything you might want to add, so I'm conscious that Sveinung's doing the majority of the speaking so far.
So I think one interesting thing could be discussing a little bit the time schedule or time expectation of the activities in the working group. So maybe this would be a nice add-on also to communicate to members or prospective members. So when we plan to meet the next time, and for maybe just for the first deliverable, when we hope to have the first results to be shared. I think we had discussed this once.
Yeah, so we have a Gantt diagram. I didn't put that up here because there was plenty of content, I thought. But I guess we could find that from it's linked from the case statement.
Yeah, on the
but
very last.
basically,
Yeah.
so the working group time here is 18 months, and that's the standard, I think, of a working group in RDA. Oh, here it is. That's great. Thanks. And the first part here would be on focusing on the metadata model, and we're aiming that to be finished within half a year. So we're starting now in June. So before Christmas, we hopefully have a first version of a data model. And then we would move towards the infrastructure parts after that, according to the plan, at least.
Okay, so month 0 would be April?
No, June, I think, because we're still not starting. Yeah.
Okay. Right. So June, and then month 5 when we have the first deliverable. So the first deliverable and all the work that will output the deliverable will be basically an harmonization of also of the things that we described today, right? Because as you said, there is a very tight common conceptualization in the presentation that I made and in the FAIRtracks schema that you presented, Sveinung. So I think for the metadata models, these two would be the main inputs, if I'm not mistaken. Of course,
yes,
there will be
a
review.
And also other review of other, but I think we have also done that. I think several of us have done that independently, a review of different models also. So we need to harmonize those reviews also, I guess.
Yes, indeed. Okay. Yeah, this seems feasible for the end of the year. I agree. So
there is a question in the chat now, or a comment from Veronica. So yeah, thank you for complimenting. So she's interested in the ethical use of the data. That's interesting. So can you say some more about the background there, what you're thinking about specifically?
Or maybe I can add something just to see if I understood correctly the hint from Veronica. So in my experience, we only worked with open data, which doesn't mean that, of course, the models and the methods that would be proposed would only work for open data. But the idea, for example, the Cancer Genome Atlas has a lot of open data and a lot of control data. But the idea is that once you stay at the level of describing the models, you can assume that the control data will somehow behave can be represented using the same formats as the open data. and that is the proposal. So I think this could partly answer. The second part is that there may be, sometimes there is a confusion between fair, a data being fair and a data being open. So fair just means that the policies and the protocols to access the data are well defined. The access policies are well described, but not necessarily the data will be openly available to everyone. So this is a second angle that we may use. So in terms of ethical, I'm also not sure about what you mean exactly.
So,
because of course, once you look
so
at
I think
yeah.
Veronica is unmuting. So
oh, perfect. Thank you.
Please provide.
No,
Yeah.
really, thank you very much indeed for the clarification. And I agree completely with your explanation. However, I was thinking more in the sense of when the users use the data which is already available from databases, and then make a publication and submit them, I mean, as original article, yeah? And I came into the, because I have, a lot of experience as being also associate editor of a journal and so on, and also getting many articles, either as editor or also as reviewer, and particularly since oftenly the authors are not familiar with the field, they are handling that data, and the output is really pretty much misleading, yeah, when we are dealing, because I'm talking about the biomedical field, yeah. The biomedical field is very sensitive in the sense that it's not enough that you say something, because what you are saying has very big implications. I mean, not only for the scientific community, but also because it's dealing with and making proposals and suggestions, yeah, and those type of articles and so on are many times used as a source for pharmaceutical companies, and you understand. So if the people has exactly as you mentioned, access to data, and they don't have the proper knowledge, I mean, to make the right interpretation and so on, it's pretty much very risky. And actually we have really an exponential growth of those type of publications, which are really making a very big mess in the biomedical field. So actually I was wondering, and particularly because also you mentioned before and so on, that you are also interested in collaborating with other groups within the RDA and so on. And I also presented this this topic because it's something that tackles many different angles, yeah, and particularly your working group is very suitable for this, because you are dealing also with genomic data, which is also, I mean, also affecting other areas, but it's also in the biomedical field. And it's very important, I mean, for the scientific and the medical point of view. But also there are other working groups, like, for example, the generative AI, yeah, which is also the other component, which is also pretty much linked to this topic, and it's very interesting because when I make a comment quite different, because it was not so focused on this type of data, but nevertheless, basically generative AI is generating, yeah, the content that people are using, I mean, to build a story and finally make the article. You understand? Because you cannot sell an article only with tables. Yeah. So they need to generate the content for the paper, which is not coming from knowledgeable people in the field, but is people which they are using generative AI, yeah, which is undisclosed, particularly to make something like sounds like a paper, which is misleading due to the lack of proper knowledge from the biomedical field, for example. And it might happen also in other fields, but I'm pretty much acquainted with the biomedical ones. That's the reason that I mention it here, because you also are dealing with genomic data, and it's very much suitable also to be included a cooperation also with this working group.
I think it's.
May we ask
Can I comment also here? Thank you very much for your discussion here. I think it's very interesting. The way I see it, this is also a quite broad topic, right? So this is a publication in general, and there's been lots of discussion of the latest years, specifically in the context of statistics. That's at least where I have been met. This is that a number of publications are actually wrong and being used for things it possibly should not be used for. But what's my point? I think as yeah, no, I remember what I was going to say. So at least where what I'm thinking, where we have been approaching previously this topic from the software point of view, and I mentioned this statistical analysis tool that we made, is that what we are typically using that for is to generate hypotheses, right? Not necessarily for concluding, and that you should always have real experiments and have some more sort of solid answers for that. But having lots of data available in a format that you can use with AI or other methods for generating hypotheses or trying to find, for instance, some connections that might be related to some diseases or something like that is important, but you should, that's not sort of the answer typically. I mean, typically you would need more rigorous analysis after that to come to some conclusion. Yeah.
I also have a comment, but maybe we can check what the chat is saying, so we involve more the participants.
Yeah, please, Anne, go ahead.
Yeah.
Yes.
Hello. Thank you for all your input presentations. I just react to the question on ethical use of the data.
Yeah.
And also in one of the presentations, there was the idea, I think it was from GH, potential adoption of from other groups, and one of the groups mentioned as a possible collaboration was the Artificial Intelligence Data Visitation Working Group, AIDV, and I'm part of this group. I just wanted to mention that within this working group, AIDV, we are in the process quite soon, I think it should be June, to open at least a number of our outputs for comments, and they are related to the ethics of artificial intelligence, and I have put the titles of those in the chat of these deliverables or outputs. There, because I thought it might be interesting. One is about consent in the context of using artificial intelligence. So it's difficult to use the classical consent form from when you use this kind of data. Then there are something about the legal foundations and also guidance for the ethics committees that will be reviewing projects or with AI or data visitation aspects. So I just wanted to mention that, because I think that related to this question of the ethical use, and specifically for genomics, that is my field, that's why I'm here. I think it might be relevant. So we are seeking for adopters of our future outputs there. So that was just my contribution.
Thank you. We're very much interested in talking more to you about this. Also now, but later. I just sort of very brief comment. So traditionally, as also when I mentioned this, is that the output, at least the public data sets, are not from particular humans. What they're typically using cell lines that's grown on lab, which makes sort of the ethics perhaps a bit simpler. But we're seeing more and more sort of single cell analysis and these kind of things, and where the individual knowledge gets more important. So that's definitely relevant. Anna, you had some comments.
Yeah, this is all very interesting, and I was wondering if at some point we stepped in some common confusion of the term FAIR, that on one side means it's an acronym for findable, accessible, interoperable, and reusable, and on the other side, FAIR means fair, right? Like that you are running AI models and you're using your data in a fair way. So we are going into the ethical concerns here. So I think on one side, this working group should nicely collaborate with those that are concerned with fairness of so using the data in a fair way, which connects with what Veronica said and also with what Anne said. On the other side, maybe could instead focus more on the idea of providing an homogenized way of representing the tracks that could work both in the case of open and closed data. And without possibly entering into the fairness problems. However, of course, then we cannot avoid the issue linked to how that data that we propose to homogenize is used. And of course, from then on, there is the whole problem of fairness. So I think we could possibly put these two things together, but being aware that they are one after the other. So in order for the data to be used, and it needs to be used in a fair way, first we want to homogenize it and make it available, findable, and clearly access with the clear access policies. That's okay. I hope.
Yeah,
no,
sure.
I agree with you. I just wanted to actually say something on the last word that you said there, the access policies. So as part of the metadata, the harmonized metadata model, I think it's very important to be very clear on what can this data be used for, and who can sort of access that. And so there's, for instance, this standard from J40H, the data use ontology, which is very much focused on that. And I'm at least thinking we probably should adopt that, but also looking at other relevant things here. But so I think that's the output. What you're providing here would be actually relevant in defining the relevant fields in the metadata model. I think that's also a bit sort of technical focus for us, but a bit concrete also.
I'm happy to kind of oh, I've frozen my laptop. Can you still hear me?
Yes,
Okay.
yes, we
Okay,
can.
cool.
Yeah.
Sorry, my screen froze for a second. Yeah, I'm happy to kind of to facilitate any connection between Anne's group and this group. So the AIDV group, actually, Anne, as you pasted that in, I was about to paste the exact same thing in. That's what another Tiger group. And I do have a message that's addressed to Anne, to yourself, and the other authors that just on the getting these deliverables boards into the process of being recognized as recommendations. I'm waiting to send that probably later today or tomorrow. But yeah, I'm happy to at some point in the future to pick that up and make that connection there. Sveinung, you're muted there.
Oh, sorry. Just before we're closing, so I was wondering to Veronica, if you're connected to another RDA group working on the ethical aspects here, or because I didn't really
Yes.
catch that.
Yeah, I think she mentioned a specific group, but I was not able to catch
Well, if Veronica can't
the
unmute,
name.
I know that she's part of this AIDV group as well. She's a
Okay,
member of
okay,
that.
so that
Okay.
might be the
It
same
might be
context
that,
then.
but there
Okay,
are
yeah.
one or two other groups, but
Yeah.
there's quite a lot of overlap in the members as well. There's a link from Federico. I think that was actually what caused my screen to freeze there for a second in the chat.
Okay, yeah, that's the data use ontology from
Nice.
J4DH. Yeah.
Okay. We are at the end of the sort of allotted time. I don't know. It seems we do have quite a small group, but I'm sure other people are busy. But if there's any sort of final questions or comments just before we move on to the very last 30 seconds, kind of actions to take away from the meeting. Does anyone have any sort of final comments or queries or questions or points to make? Okay. So I guess the next step then, what I will do, I'll just paste into the chat. So we did, Sveinung did mention, and Anna as well did mention about the sort of specifics of the meeting schedule, and Veronica, you just put it in the chat.
Yeah.
So what I'll do is I will paste this into the document here. So first thing I would encourage everyone to do is to the formatting has gone strange, but it's still legible in the chat. So please join the working group. So this will obviously give you access to put you on the kind of contacts list for the group, and we'll be able to contact the group as a whole there. We do have the regular meetings. So initially these will be taking place monthly. We have decided on the third Tuesday of each month. So the first of these will be starting in, Swan has on screen there, and starting next month on the 18th of June. So we'll be posting. I'll be posting a link to a Zoom link, and then just reminding people of that, and just any other updates on the group between now and then. But please, just going back to that first point, please sign up to join the working group. That's the best way to keep up to date. Even if it's a case that you won't be able to join at that time, or can't devote a whole lot of effort or attention at the beginning, that's the best place to just sign up and to be kept up to date. I think that is it from me. Oh, Sveinung, sorry, you have the very last slide there.
Yeah, wait, sorry, no, I got that there. Okay, can you hear me now? Yeah, yeah.
Yep.
Yeah, no, so just want to mention also that we are still looking for another co-chair to focus a bit on the biodiversity, because all of the existing ones have more of a biomedical focus. So if you know somebody or have some idea, then please let them know or let us know. And in the end, I just want to thank very much Ryan and others in the RDA Tiger project. They've helped us a lot getting these working groups together and connecting co-chairs and organizing everything. So I'm very grateful for the work they've been doing. And Ari Asmi, who then put us in the RDA Tiger application itself as a pilot. Peter McCallum from Elixir, the technical leader of Elixir. He put us in contact with Ariasmi, so thanks a lot for that. And also to thank Jeff Christensen from Australian Biocommons and also part of RDA who specifically helped me a lot in a trip I had to Australia last year trying to promote this working group. So I think that was all I had.
Okay.
Thanks, everybody. So thanks for joining. I guess
And
we can wrap up.
thanks to all the co-chairs also.
Yeah, of
Thank
course.
you.
And hopefully we'll see you
Thank you.
in some meeting soon, or if not, at least on the list of members of the group.
Yeah. So we'll see each other on the 18th of June then.
Yep. If not before.
Okay.
Thanks, everybody.
Okay. Yes. Thank you. Bye-bye.
Bye.
Have a good day.